Gene identification in prokaryotic organisms is simplified by their lacking A. Exons, B. introns, C. coding segments, D. useful nucleotide sequences. The correct answer is introns. Screening the predicted protein sequences against library confirms the prediction and expression of the gene. A. Expressed sequence tag EST. B. Tags. C. Palindromes. D. Proteomes. The correct answer is expressed sequence tag EST. In case of genome sequence assembly, which of the given statement is incorrect? A. Full chromosomal sequences are assembled from the overlaps in a highly redundant set of fragments by an automatic computational method or from the fragment order on a physical map. B. Chromosome cloning is carried out in bacterial artificial chromosomes banks. C. Chromosomes of a target organism are purified, fragmented, and subcloned in fragments of size hundreds of bp. D. Genome sequences are assembled from DNA sequence fragments of approximate length 500 bp obtained using DNA sequencing machines. The correct answer is chromosomes of a target organism are purified, fragmented, and subcloned in fragments of size hundreds of bp. Which of the given statement is incorrect about functional genomics? A. Functional genomics involves the preparation of mutant or transgenic organisms with a mutant form of a particular gene, usually designed to prevent expression of the gene. B. An abnormal properties of the mutant organism does not reveal the gene function. C. When two or more members of a gene family are found rather than a single match to a known gene, the biological activity of these members may be analyzed by functional genomics to look for diversification of function in the family. D. A more detailed analysis of the relative amount of sequence variability in a chromosomal region within populations of closely related species can reveal the presence of genes that are under selection. The correct answer is an abnormal properties of the mutant organism does not reveal the gene function. Which of the given statement is incorrect about the gene map? A. Gene order in two related organisms reflects the order that was present in a common ancestor genome. Chromosomal breaks followed by reassembly of fragments in a different order can produce new gene maps. B. Gene order is only revealed by the physical order of genes on the chromosome. C. Sequence variations, polymorphisms that are close to tightly linked a trait may be used to trace the trait by virtue of the fact that the polymorphism and the trait are seldom separated from one generation to the next. D. These types of evolutionary changes in genomes have been modeled by computational methods. The correct answer is gene order is only revealed by the physical order of genes on the chromosome. Each genome sequence is scanned for protein-encoding genes using gene models trained on known gene sequences from the same organism. A. True. B. False. C. D. The correct answer is true. Sequencing of genomes depends on the assembly of a large number of DNA reads into a linear contiguous DNA sequence. A. True. B. False. C. D. The correct answer is true. These transposable elements can at most comprise 1/4 of the genome sequence. A. True. B. False. C. D. The correct answer is false. Which of the following information is not directly obtained by microarray analysis? A. Which genes are expressed at a particular stage of the cell cycle? B. Which genes are expressed at a particular stage of developmental cycle of an organism? C. Which genes are depleted at what time? D. Genes that respond to a given environmental signal to the same extent. The correct answer is which genes are depleted at what time.